Welcome to Drugs and Dermatology. In this video, we will be discussing the current FDA-approved anti-interleukin-23 agents relevant to dermatology. Guselkumab, known as Tremfya, tildrakizumab, known as Ilumya, and risankizumab, known as Skyrizi. An important note is although this class of drugs may be utilized for a wide range of treatments, we will be looking through a dermatology lens focusing on dermatological diseases that are FDA approved. So, let's break it down, drugs in dermatology style. The interleukin-23 P19 inhibitors are a selective upstream approach to targeting the interleukin-23 Th17 axis. Whereas ustekinumab blocks both interleukin-12 and interleukin-23 through their shared P40 subunit, these agents target only the P19 subunit, a component unique to interleukin-23, leaving interleukin-12 signaling and Th1 host defense intact. Guselkumab, with the brand name Tremfya and developed by Janssen Biotech, was the first selective interleukin-23 P19 inhibitor to receive FDA approval. It is a fully human IgG1 monoclonal antibody administered by subcutaneous self-injection. It has a demonstrated superiority over adalimumab in the Voyage 1 and Voyage 2 trials, and superiority over secukinumab in the Eclipse trial. Guselkumab has also shown efficacy in difficult-to-treat anatomical areas, including scalp, nails, and palmoplantar psoriasis. Risankizumab, with the brand name Skyrizi and developed by AbbVie, is a humanized IgG1 monoclonal antibody also targeting the interleukin-23 P19 subunit. It has demonstrated superior efficacy to ustekinumab in Ultima 1 and Ultima 2, to adalimumab in the INVENT trial, and to secukinumab in the IMHANCE trial. It offers the longest maintenance dosing interval in the class at every 12 weeks. Tildrakizumab, with the brand name Ilumya and originally developed by Almirall and Sun Pharmaceutical, is a humanized IgG1 monoclonal antibody targeting the same P19 subunit. It was evaluated in the Resurface 1 and Resurface 2 trials. It is also uniquely clinician administered rather than self-injectable, which distinguishes it from other interleukin-23 inhibitors. The development of the selective interleukin-23 P19 inhibitors was a direct scientific descendant of ustekinumab's success. Once it became clear that blocking interleukin-23 rather than interleukin-12 was likely responsible for the majority of ustekinumab's therapeutic effect in psoriasis, the field transitioned towards agents that could achieve this more selectively. Guselkumab was the first to receive FDA approval on July 13, 2017, for moderate to severe plaque psoriasis in adults, making it the first selective interleukin-23 P19 inhibitor ever approved. In July 2020, the FDA approved guselkumab for active psoriatic arthritis in adults based on the Discover 1 and Discover 2 Phase 3 trials. Additional non-dermatologic approvals for ulcerative colitis and Crohn's disease followed. Tildrakizumab received FDA approval on March 21, 2018 for moderate to severe plaque psoriasis in adults, becoming the second selective P19 inhibitor approved, approximately 8 months after guselkumab. It has received no additional FDA-approved indications to date. It remains the only agent in the class without a psoriatic arthritis indication. Risankizumab received FDA approval in 2019 for moderate to severe plaque psoriasis in adults, making it the third P19 inhibitor to be approved. In January 2022, it received FDA approval for active psoriatic arthritis in adults. Additional non-dermatologic approvals for ulcerative colitis and Crohn's disease followed. Guselkumab holds 2 FDA-approved dermatologic indications: moderate to severe plaque psoriasis in adults and active psoriatic arthritis for patients aged 6 years and older. Risankizumab likewise holds 2 FDA-approved dermatologic indications: moderate to severe plaque psoriasis in adults and active psoriatic arthritis in adults. Tildrakizumab holds 1 FDA-approved dermatologic indication: moderate to severe plaque psoriasis in adults. It has no FDA approval for psoriatic arthritis or any other indication. All 3 agents share the same molecular target, the P19 subunit of interleukin-23, and the same general mechanism. The cytokine interleukin-23 is composed of a P19 subunit unique to interleukin-23 and a P40 subunit shared with interleukin-12. It is produced primarily by dendritic cells and macrophages in the psoriatic dermis and acts as the master regulator of Th17 cell differentiation, survival, and maintenance. Sustained Th17 activation results in abundant production of interleukin-17A, interleukin-17F, interleukin-26, TNF-alpha, and interferon gamma. These are the proximal effector cytokines that act on keratinocytes to drive barrier disruption and the chronic inflammatory cascade of psoriasis. By binding the P19 subunit specifically, all 3 agents prevent interleukin-23 from engaging its receptor complex without affecting interleukin-12. Since interleukin-12 signals through a different receptor complex, which drives Th1 differentiation and NK cell activation, critical for host defense against specific pathogens, the theoretical vulnerabilities to mycobacteria and salmonella associated with the P40 blockade are not a concern with P19 selective agents. The downstream consequences of P19 inhibition are the same across all 3 agents: disruption of the interleukin-23 Th17 axis, reduction in Th17 cell differentiation, and suppression of interleukin-17A, interleukin-17F, interleukin-26, TNF-alpha, and interferon gamma production. The class-wide adverse effect profile of interleukin-23 P19 inhibitors is fairly homogeneous and is very favorable. The most common adverse effects include upper respiratory tract infections, headache, injection site reactions, arthralgia, bronchitis, gastroenteritis, tinea infections, and herpes simplex infections. Notably, across all 3 agents, pooled clinical trial data and real-world data have shown no cases of progression from latent TB to active TB. Furthermore, inflammatory bowel disease has not occurred with any of these P19 interleukin-23 inhibitors. The only absolute contraindication for all 3 agents is a history of serious hypersensitivity reaction to the drug or any excipient in the formulation. Shared warnings and precautions include pre-treatment evaluation for TB is mandatory for initiating any of the 3 agents, and treatment of latent TB should be completed before initiation. Although pooled data have not demonstrated progression from latent to active TB in clinical trials, screening remains the standard of care. live vaccines must be avoided during treatment with all three agents. Pregnancy safety is unknown for all three agents, and as IgG antibodies, they are expected to cross the placenta, particularly in the third trimester. Unlike the interleukin-17 inhibitors, none of the P19 inhibitors carry an inflammatory bowel disease contraindication. As always, we will close our video with a table going over the main talking points. The interleukin-23 inhibitors we discussed were guselkumab, risankizumab, and tildrakizumab. I hope you have a better understanding of anti-interleukin-23 therapies. This is meant for educational and informational purposes only and should not be used as a substitute for medical advice, diagnosis, or treatment. Thank you for watching, and see you in the next video.